Welcome to Vital Biology classes. For new updates, subscribe to the channel Vital Biology. The theme of our channel is Study Now, Be Proud Later. You can join our Vital Biology Telegram group and all the classes, notes, PDFs are available there. Link is provided below in description box. Hello everyone. This class is for all the Kset aspirants, and it's also helpful for KCET too, KCET biology, but it's best and most important for the Kset license itself. Why? Because some of this information is not included in the KCET syllabus, but in Kset this all the points and topics are very much important and it is included in the Kset syllabus. Okay, so Kset aspirant should listen these things carefully. So in the last class we have studied about the size of the cell in human beings, right? There we completed that sperm cell, that is smallest cell, and it's also motile cell, and the largest cell, that is ovum cell, present in the female, and the longest cell, that is nerve cell, that is up to 1 meter, that is also present in human. We studied this much. So let us move on to the next slide today, that is about the shape of the cell. Okay, shape of the cells. So here the shape of the cells varies according to their, based on their function. Okay, here they have given that cells vary in shape. Why? Because variation depends mainly based on upon the function of the cell, how it functions. Based on that the shape of the cell is dependent. And next thing is, for example, let us take the euglena and amoeba. They just beyond changing their shape, means totally we can tell that irregular in shape. Okay, so euglena and amoeba can be changing their shape, and most of the cells does not change their shape. It just have a fixed shape. Most of the cells have a fixed shape. Okay, so I think you guys have understood these 3 points, right? So let us move to the another points. Those are the 3 examples based on the cells they have given. That first is human RBC, and second is nerve cell, and third is human white blood cells. Okay, so here let us go to the first one, that is human RBCs. So here RBCs is also called as red blood cells, and it's mainly called as erythrocytes too. For example, one of the question was arised in one of the year, that what means like RBCs is also called as options: leukocytes, thrombocytes, erythrocytes, none of the above. So here you want to answer that erythrocytes. RBCs is also called as erythrocytes. Okay, so here as I told you, the size is based on their function. A cell ಹೆಂಗೆ ಫಂಕ್ಷನ್ ಮಾಡುತ್ತೋ ಅದ್ರ ಮೇಲೆ the shape of the cell is dependent. Okay, so here this shape of the cell, RBC cell ಹೆಂಗಿದೆ ಅಂದರೆ circular and biconcave. Okay, why these cells are circular and biconcave? Because ಈ shape ಇದ್ದರೆ, means circular and biconcave ಇರೋದ್ರಿಂದ these cells can easily pass through the human capillaries. So like blood supply aguttalva, so those are called as capillaries. So in that it will just pass easily and fastly, so that it can share and supply the oxygen and other nutrients and minerals to the ATPs to the other organisms. Okay, so it has a circular and biconcave. Okay, and second one, that is nerve cell. Okay, second one, that is nerve cell. Nerve cells are branched to conduct the impulses from one point to the another point, or else from one nerve to the another nerve. Okay, so here I'll draw a simple structure. oh sorry, this is, let us take this as an one example. Okay, that is one neuron. I think when I'm drawing you can understand that, oh, is it this? I have seen already, right? So this is axon. Okay, and another neuron, I'll draw it here. Okay, these thread like structures are dendrites, called as dendrites. This is again axon. So here this dendroid, just connect this diagram too with this. So this dendroid, this second dendroid, and this is third, and sorry, this is first nerve, second nerve, and third nerve. Okay, this is third nerve. So from first nerve it is getting the information here through dendroid. Okay, this first nerve, it is supplying the information from that first nerve to the second nerve, so that it just take the information from here through dendroid to here. Okay, and again from here this second nerve supplies the, or else transfers the information again from here to one of the dendroid. Okay, one of the dendroid, it takes the information from here and it again passes to this, and again another neuron arises and it just passes there. So here the impulses is nothing but this connection where the information or else message that is passing from one nerve cell to the another nerve cell, that is called as impulses. So this is a dendroid, as I told you, and this is axon, simple thing, and the information is passing from here to here. Okay, so that connection and that message is called as information, passing the information is called as impulses. Okay, it's more than enough, because in later classes I'll be discussing about this in a deep thing, but it's a basic class, so just I'll stop it here, means about explaining in a deep way. And this is all about neuron. Okay, I guess you have understood it. And next let us move on to the third, that is human WBCs. WBCs is nothing but white blood cells and can change their shape to engulf the microorganisms that enter the body. Okay, like engulf is nothing but eating. Okay, eating or drinking, engulf. Sorry, one second. Engulf, nothing but eat or drink. So ಏನ್ನ eat ಮಾಡುತ್ತೆ and ಏನ್ನ drink ಮಾಡುತ್ತೆ means microorganisms. Okay, it eats or drinks the microorganisms which infects the living body. Okay, for example, so let us take this is one of the microorganism that it is infecting. Okay, that it is infecting the human body. Okay, it is infecting the human body. So in human body, like there is WBC, right? So what this WBCs do, let's see. So as I told you, this is a microorganism, and let us take this as a white blood cell. Okay, white blood cell is actually white in color, but if I select the white color here, you can't see. Okay, so this is a white blood cell. It also contains a cell. So white blood cell, what it does means it just engulfs means, as I told you, it eats the microorganism. When it is eating the microorganism, it just changes the shape. Okay, finally what it does means, so it eats the, finally it eats the microorganism, and first it was shaped as like in this way, right? And finally when it is eating the microorganism, it just changes the shape and finally it becomes in this way. Okay. Because of its white blood's function itself, the size is based. I guess you have understood clearly about all these three examples, that is RBC, nerve cell and white blood cells. So another thing, white blood cells is also called as leukocytes. Why it is called as leukocytes means, here white leuco means white in color. Okay, the white color is, it's also called as leuco. Okay, and cytes is nothing but cell, white blood cell or else leukocytes, white cell, leukocytes. Okay, let us move on to the next one, next slide, that is structure of cell. Okay, let us learn about the structure of cell. So in structure of cell, the detailed structure of the cell has been studied under the compound microscope and electron microscope. So the detailed structure, it may be any mitochondria, endoplasmic reticulum, and like Golgi complex, lysosomes, ribosomes, cytoplasm, cytoskeleton, everything. This detailed structure, what and all it contains, that is studied under the compound microscope and electron microscope. Okay, certain structures can be seen only under the electron microscope, as I told you. For example, like mitochondria or else endoplasmic reticulum and Golgi complex. Okay, these type of structures are only seen under the electron microscope. Okay, so the structure of the cell has seen under the electron microscope is called as ultrastructure. Why means, ultrastructure means we can see very like anatomically, means very depthly we can see under the electron microscope. Okay, so it is called as ultrastructure. Okay, so let us see the compound microscope. So compound microscope magnification is just 2000x. Okay, magnification is just 2000x, and electron microscope's magnification is 500,000x. Okay, 500,000x, just remember it. So these two microscopes that gives a detailed structural information about the cell. Okay, next let us move on to the next slide, that is let us take a animal cell. As you know, there are three types of there are three types of cells, that is animal cell, and another one is plant cell, and another one is bacterial cell. Okay, bacterial cell. So here these two are called as, means animal cell and plant cell is called as eukaryotic cell, and this bacterial cell is called as prokaryotic cell. Okay, so first let us see the animal cell. Animal cell, you can observe here, this is a animal cell structure. So here in the animal cell structure you can observe there are several organelles and parts and fluid like structures. And can you name this animal cell structure without seeing the parts in the left side? Okay, first test it yourself for a few minutes. Pause the video and stop the video and just guess how much you can remember the organelles. Okay, so afterwards you can continue the video. So I'll just continue with it. So this first one is nucleus. Okay, you can observe here, this is a nucleus which carries a genetic material. Okay, which carries the genetic material. This is a nucleus. Okay, and next second one, This is Golgi body. Okay, you can observe here this green color structure, that is Golgi body. Okay, that secretes the chemical and proteins, lipids in this way. And the third one is vesicles. Okay, vesicles function is just like it takes some microorganisms which infects and just throws out of the cell, and some of the proteins or lipids, it's very like any important elements that is very useful to the cell in anything like development, anything, it also engulfs. Okay, now here you can see that image that like the vesicle is here, this vesicle is throwing the infected particles or the unwanted substance or waste substance that it is throwing out of the cell, right? You can observe here. So here is also one that is throwing out unwanted substance. And next let us move on to the fourth, that is this outermost layer in the animal cell is plasma membrane. Okay, when you see in plant cell, the outermost layer is cell wall, and when you see in the bacteria, the protective layer is capsule, and the second layer from outside if you come means the first layer is capsule and second layer is cell wall and third layer is plasma membrane. Okay, but in animal cell the cell wall is absent, so outermost layer is plasma membrane. Okay, and let us move on to the next, that is fifth part, that is mitochondria. This is mitochondria. Okay, mitochondria, that functions, its function is to give a ATP, energy. Okay, and it's also called as powerhouse of cell. Okay, and let us move on to the sixth part, that is cytoskeleton. So for example, this is a cell, take it as a cell. So in cell you can observe that something here and all, nucleus, Golgi complex, endoplasmic reticulum, but you can also observe that like something skeleton like structure, right, mash like structure, in this way it will be. So this is called as cytoskeleton, which helps to organelles to get some messages from one organelle to the another organelle. So it's very helpful for cell in the name, that is cytoskeleton, and it also helps in the structure to maintain the particular structure of the cell. So this cytoskeleton is helpful. And let us move on to the next organelle, that is centriole. Centriole is very important and helpful, and it's only present in the animal cell. This centriole is helpful, and how it functions means it helps in the during the cell division, that is mitosis and meiosis. During that time the centrioles are very helpful, and it's a very important thing which produces the spindle fibers. Okay, when you study the cell divisions, you will know that the spindle fibers that carries the chromosomes when in the phase of anaphase stage. Okay, in the phase that is anaphase, by the time these spindle fibers are produced by the centrioles, and these spindle fibers just carries the centrioles and divides the cell into two. Okay, its main function is that, and in coming slides I'll be explaining it in a very deep way, but just it is, as I told you, this is just a basic class. Okay, and next, eighth part, that is lysosome. Lysosome, it is also called as suicide bags. As you know, in childhood classes in 8th, 9th, 10th, it's a very important lesson, that is cell. Okay, so lysosomes is also called as suicide bags. Why it is called as suicide bags? It means like its main function is to digest. Okay, digest the unwanted things in the cell, and sometimes if that particular this lysosome itself it's not working properly, then it itself engulfs, and it just destroys itself. Okay, so they are called as suicide bags. That itself destroys itself when it is not used for the cell. Okay, so it is called as suicide bags. And let us move on to the next, that is ninth part, that is cytoplasm. So cytoplasm, it's nothing but like a fluid like structure present in the cell that carries all the organelles, okay, that gives some nutrients and proteins, lipids for all the organelles, and it supports to float or maintain the particular places for those organelles, okay. Cytoplasm function is that. And next 10th one, that is rough endoplasmic reticulum. Rough endoplasmic reticulum is mainly used for the synthesis of protein. Why? You can think that ribosomes are the main organ that synthesizes protein, right? So you can think in that way. Yeah, that's right. Why? Because here on the rough endoplasmic reticulum the ribosomes are present, and hence it is called as rough endoplasmic reticulum. Why? Because if there is a simple line, it is called as smooth, or else if there is some dots means it's rough, right? So in this way the endoplasmic reticulum, half of the endoplasmic reticulum is seen, so it is called as rough endoplasmic reticulum. And another one is there, 11th part, that is smooth endoplasmic reticulum. So smooth endoplasmic reticulum is as I told you, just it is like a simple line. It there is the absence of ribosomes, okay. So in smooth endoplasmic reticulum there is no any synthesis of protein, but there is the synthesis of lipids in smooth endoplasmic reticulum. In rough endoplasmic reticulum there is a synthesis of proteins, and at any cost this rough endoplasmic reticulum does not synthesize the lipids, and at any cost the smooth endoplasmic reticulum does not synthesize the protein. It just have its own function, that smooth endoplasmic reticulum synthesizes the lipids, and rough endoplasmic reticulum just synthesizes the proteins. Okay, next let us see the 12th part, that is nucleolus. Nucleolus is present inside the nucleus, okay, that carries all the genetic material, yeah, that is DNA. And next from the DNA it may forming the chromosome protein, and finally it makes chromosomes, okay. So these are the genetic material that are present inside the nucleolus, okay. So today let us stop this class here itself, and in the next class let us continue the things, okay. Thank you. Bye bye. Thank you. All the class notes and other study materials provided in our Vital Biology Telegram group. Link is provided below in the description box. Like, share, subscribe and press on the bell button for the notification of newly updated classes.